evening. My name is Shinyi Pai, and I'm the Program Director for Town Hall. On behalf of the staff at Town Hall Seattle and our friends at Third Place Books, it's my pleasure to welcome you to tonight's talk with Bill Schutt. As we get underway, I would like to acknowledge our institution stands on the unceded traditional territory of the Coast Salish people, particularly the Duwamish. We thank them for our continuing use of the natural resources of their ancestral homeland. We're so glad to have you join us tonight. The presentation will run about 60 minutes, including Q&A. To submit your questions for the Q&A portion of the event, please enter meet.ps/Schutt or scan the QR code right now on the screen with your smartphone. We'll drop this link in the chat and remind folks where to go when we get to Q&A. We can't guarantee that we'll be able to address every question, but we'll try to get to as many as possible. You can help us by keeping your own question concise. For those who would like to view the program with closed captions, click the CC button in the bottom right corner of the video player. Town Hall is adding new events and podcasts every day. Upcoming events include Michelle Miller-Fisher, Amber Winnick, and Zoe Greggs on designing motherhood, things that make and break our burrs, and Bartow J. Elmore on Monsanto's past and our food future. Visit our website to join our email list and get the latest updates as more programs are added throughout the season. Town Hall's work is made possible through your support and the support of our sponsors. As part of our Arno G. Matulski Science Series, this event is supported by Microsoft and Wyncote Foundation Northwest. Finally, Town Hall is a member-supported organization, so I'd like to thank all of our members who are joining us tonight. If you share in Town Hall's vision of a community energized and empowered by considering questions of politics, science, and culture, please consider supporting us by becoming a member. You'll find membership information on our website. Lastly, you'll definitely want to dive deeper into this evening's topic by purchasing a copy of Schutt's book. The quickest way to do so is to visit the link in the chat below, which will take you directly to our friends at Third Place Books. Bill Schutt is a vertebrate zoologist and author of 6 nonfiction and fiction books, including the New York Times Editor's Choice, Cannibalism, A Perfectly Natural History. Recently retired from his post as professor of biology at LIU Post, he is a research associate at the American Museum of Natural History, where he has studied bats all over the world. His research has been featured in Natural History magazine, as well as in the New York Times, Newsday, The Economist, and Discover. His new book, Pump, A Natural History of the Heart, is the subject of tonight's talk. Please join me in welcoming Bill Schutt.
Good evening. It's really nice to be here tonight, and especially to talk about my new book, Pump, A Natural History of the Heart. I was thrilled this morning when I woke up. I got a message from my editor and my agent that the Seattle Book Review had given Pump a 5-star review. So that was kind of thrilling and kind of neat that I'm going to be giving a talk tonight to you folks. So yes, as just mentioned, I retired about a year ago. I taught anatomy and physiology at Long Island University. And I've been a research associate for years at the American Museum of Natural History. Before that, I was a postdoctoral fellow there, and before that, a graduate student. So one thing I'd like to talk about before I move on is that the illustrations that you see are by my longtime friend and a wonderfully talented artist, Patricia J. Wynn, who has pretty much illustrated every scientific paper that I've ever written, every book chapter and every novel, and all 3 of my nonfiction books. So I just want to give her a shout out. So my first book was Dark Banquet, Blood and the Curious Lives of Blood-Feeding Creatures. And that sort of came about because I studied vampire bats for 20 years. And so I got this opportunity to work on a popular science book. And it made sense that I did this. I've always been into the macabre, and I really wanted to take this topic, vampirism and blood feeding, and demystify it. And luckily for me, but unluckily for a lot of people, things that were taking place at the same time I was writing this book were the problems that were out there, especially in cities, with bed bugs, and also Lyme disease with ticks. And so the first half of the book was about me and the work I did on vampire bats with a lot of other folks. And the second part of it moved into all of these kinds of strange behaviors and that were out there in the animal kingdom, And the follow-up to that made sense to me. It was cannibalism. As I said, I've always been into sort of strange topics, and nobody's really too, people who haven't seen me in 20, 30 years, and then they contact me through social media, no one is really surprised when they find out that I spent most of my career working on vampire bats. And so it was no stretch to move into cannibalism. And once again, I found this niche between the sort of academic work on one side that only scientists would be interested in reading, and on the other side, sort of sensationalized stuff. And I found this spot in the middle where I was able to go in and take a topic like cannibalism and get rid of the jargon and explain it in a way where I was able to go in and talk about other topics as well, other that I thought were related and important. And so I was really lucky. Cannibalism did quite well. I've been having interviews with that book since it came out in 2017. So given the fact that Dark Banquet was about vampirism and cannibalism was about cannibalism, how did I come to write a book about the heart? And really what took place is, as I was thinking about the book, what book I wanted to work on next, my editor and my agent both suggested that I take a look at some topics that might be more mainstream for my next book. And so one of the things that they gave me in this list was the heart. And initially I thought, well, this has to have been done before. And there are so many books about the heart that how am I going to go in there and tell the stories that I want to tell? And it didn't take me very long to figure out that there was room for this type of a book. And the more that I looked into it, moving through the animal kingdom, as you'll see, the neat examples that I found turned out in many instances to have scientific relevance today in the field of cardiac medicine. So the learning curve was steep, just as it was in my first two books. But I think I found enough neat history and stories about animals, and then the tie into to ancient and modern medicine, and then medicine of the future to satisfy my readers. And I hope that you'll enjoy this sort of summary that I'll be giving this evening. So I'm going to start off with a little bit of a lesson here, and that's, what are hearts and circulatory systems? That's what this book is about. And the short answer is that, and for my money, we're talking about muscular pumps that move a fluid like blood around the body through a system of vessels. And the function of that is to carry oxygen and nutrients, carbon dioxide, and waste products. Now you'll notice there's the star there, not insects. As we're going to see, the insect circulatory system does not carry those gases, oxygen and carbon dioxide. It is solely deals with nutrients and waste products. Now, do all organisms need hearts and circulatory systems? The short answer to that is no, not at all. And who doesn't need a circulatory system? Who doesn't need a heart? These are creatures that are extremely small, or they're really, really flat, like sponges, anemones, corals, starfish, things like that. And of course, single-celled protists. And so you might ask, well, why? What is it about these creatures that allows them to not have a muscular pump and the vessels that go along with it? Here's the deal. If you're really tiny, like this single-celled organism here, you generally get your oxygen by going into an environment that has a higher concentration outside of your cell than inside. And so that oxygen, in this case, just moves I'll move to the next, from a high concentration outside the cell to a lower concentration inside the cell. And that takes place by diffusion. Now, by the same token, a product of the cell doing its thing is that it produces carbon dioxide as it is converting energy of nutrients into a form of energy that it can use. So carbon dioxide is a waste product, and if it builds up, it can be toxic. So there's a higher concentration of carbon dioxide in the cell than there is in that outside environment, and that carbon dioxide just diffuses out. And by diffusion, I mean that it goes from a higher concentration to a lower concentration. It's following its concentration gradient, and it just passes through this porous cell membrane. So if you're really small or really flat, you don't need a circulatory system. But in order for creatures to have evolved any type of size at all, And we're talking about now animals that are millions, that are composed of millions of cells and many cell layers thick. Diffusion does not work very efficiently for these types of animals. It works very slowly, but not fast enough to be able to move oxygen, carbon dioxide, nutrients, and waste products when you are talking about something that has a large size. So large size animals could not evolve unless a system evolved that enabled these materials that I've just mentioned, oxygen, carbon dioxide, waste products, and nutrients, to move them around through the body, to pump them around through the body, through this cardiovascular system that many of us are familiar with. Now, that's not to say that there are not that there's not a tremendous amount of variation, because there certainly is. Now, if you look, for example, in the invertebrates, these are animals that do not have a backbone. Many of them, like this insect that you see here, have what are known as open circulatory systems. And what that means is that the heart, and here I'm putting the heart in sort of quotation marks, because if I were to show these structures to a cardiac surgeon, he would laugh at me and tell me that's not real. That's not a card carrying heart. It doesn't have the lining that you need for me to consider this thing to be a heart. But what it is a muscular pump, and I'll show you in a minute how that works. But in any event, an open circulatory system means that at some point, the liquid that's being pumped by this heart is going to leave the circulatory system and enter into a chamber called a hemocoel. And once it's in that chamber, it then bathes the cells that surround the chamber and supplies them with for example, nutrients and takes away waste products. As I said before, when you're talking about insects, oxygen and carbon dioxide are carried in a very different way. Now, a closed circulatory system, which is what we're probably more familiar with, is a system where when the blood is pumped, it does not ever leave the circulatory system. So these are going to be, for the most part, animals like vertebrates. But as you're going to see, this is also, closed circulatory systems are also found in some invertebrates. Here you can see that closed system in a fish with its single atrium and ventricle. This is the receiving chamber. When I say atrium, think this is a chamber of the heart that receives blood. And the ventricle is, when I say ventricle, I want you to think of pumping chamber. This is the chamber that's going to pump blood out of the heart. Okay, so how does a heart pump blood? When you're looking at our heart, what you can see here is that there are literally whorls of muscle tissue that form the walls of these four chambers. You have a right atrium, a left atrium, a right ventricle, and a left ventricle. And of course, so if you can sort of think about this as, if my hands are these muscle fibers that are surrounding this chamber, when those muscles contract, the volume inside that chamber is decreased, and the blood is going to move from place to place. If the atria contract, it moves into the ventricles. If the ventricles contract, it moves out of the heart. Now, where does the stimulus for that contraction of muscle come from? In hearts like the human heart and mammalian hearts, and many other hearts but for that matter, the stimulus for that contraction comes from inside the heart itself, in specialized tissue called the pacemaker. So, and we're pretty much familiar with the fact that if this pacemaker starts to break down, you put in an artificial pacemaker, and that sends out this kind of similar electrical signal that passes through the heart. And as it passes through the heart, the ventricles, the atria, and then the ventricles contract in the wake of that signal. Okay, so that's pretty much a simplified version of how this works. Now, but there is an assist to the heart now. And I want you to look here at, so this is the aorta. So when your left ventricle contracts, the blood is going to leave the aorta, is going to leave the ventricle and enter into the aorta, and then be pumped out to the body. Keep that in mind. Okay, so, and here's actually a better picture of it. In invertebrates like us, the heart is separated in mammals especially, and in birds and some reptiles, the heart is separated into really two sections. So it's a double pump. On the right side, you've got blood returning from the body. It's deoxygenated now, and it enters into the right side, and the right side pumps it to the lungs, picks up oxygen, gives up carbon dioxide, returns to the left side. It's now oxygenated. It's now depleted in carbon dioxide, and that left side pumps it to the body. Okay, so just a review. You've probably seen this stuff many times. But there is an assist, as I was mentioning before, and there's that aorta again, right there. Now, picture that you have your hands wrapped around a water balloon, and that water balloon is full of water. And your fingers are these muscle fibers. When they contract, and this is a vessel like the aorta, what would happen is this: you squeeze, and now you're causing that volume to decrease. You're increasing the pressure inside there. And what's going to happen? The water is going to move, right? Well, the same thing is happening in large vessels that are found just outside the heart, like the aorta. So it gives an assist to the heart in a way that allows it to help it pump blood. So when you hear about things like hardening of the arteries, just think in terms of if this structure were rigid and did not have this ability to contract, that causes the heart to be stressed, and that can cause serious problems. So just a bit of general information for how these types of things work. All right, so let's talk about invertebrate hearts for a while. And a key message here is that there are many, many different types of invertebrate hearts and circulatory systems. And the reason for that is that we believe that in these animals, circulatory systems and hearts evolved many times. So there's all sorts of variation that you see here, from the fact that you have a closed system in squid that has 3 hearts. It had 2 hearts here, these brachial hearts, pump blood that's returning from the body, deoxygenated to the gills, and then that blood enters a systemic heart, which pumps that blood to the body. So very strange, but a closed circulatory system in this invertebrate, probably because of their extremely active lifestyle. The fact that they're very, very intelligent, they're fast moving, so they require a great deal of blood. Something like an insect is kind of strange because it has multiple structures that one might consider to be a heart. And so it's got tiny little heart-like structures that, for example, pump hemolymph, which is what the liquid is called in insects, to the antennae, others to the legs. I always sort of put out an alarm here, or an alert, because not a lot is known about those particular types of hearts. So it's a great place for graduate students to go in there and figure out a project. I mentioned before to you that the hemolymph, or the equivalent of blood in insects, doesn't carry oxygen and carbon dioxide. That enters through the holes in the side of the body, which you can see here, called spiracles. And that air, which carries oxygen, moves into a vessel and then breaks off into smaller and smaller and smaller vessels until it's able to supply air to tissues and organs. Now, this is in the book. I talk about the fact that Mothra, one of my favorite monsters from childhood, could not exist because this system works perfectly well for small creatures like insects, but it could never support a Zeppelin-sized moth. So Mothra, in actuality, couldn't exist with the type of respiratory system that insects have. Here, everyone has dissected a worm as either in science class or maybe in high school. But in any event, they've got these 5 structures here called aortic hearts or aortic arches, and they get the job done as well. They also get a lot of their oxygen right through their skin. If you've ever seen worms out on sort of a wet night, they're being sort of driven out of the ground, but they're also getting oxygen and giving off carbon dioxide through their skin. A key message here is that and I want my readers, I really wanted my readers to appreciate this, is that just because, we have this habit of considering any type of organ or behavior that is not human to be somehow faulty or primitive or defective. Not the case. These types of systems, though they may be somewhat simpler than what we have been evolving for millions and millions of years, and they do the job that they evolved to do just fine. So we really need to move away from this idea of the fact that, our heart is the best heart. Well, our heart's very complex, and that complexity leads to problems. So it's not really the best way to think about structures like that. These are different, but they work perfectly fine for the creatures that own them. Okay, so let's talk a bit about vertebrate hearts. When you get into the vertebrates, animals with a backbone, what you're going to see is that there are tremendous similarities between them, even between, and this is going to become important later when we talk about the fact that many of these non-human vertebrates are being used and in spectacular fashion in order to improve our knowledge about cardiac medicine. So if you look at these creatures, that they are roughly similar in the makeup of their circulatory system, because unlike the invertebrates, we think that the vertebrate heart and circulatory system only evolved once in a common ancestor of all of these creatures. And then you certainly have some differences. Fish, for example, have two chambers, a two-chambered heart. Reptiles and amphibians have three chambers, except for crocodiles and alligators, birds and mammals, which have four-chambered hearts, and that's the type of heart that most of us are familiar with these four chambers. Okay, so I'm going to tell some stories now that I reviewed, that I talked about more in depth in the book, and I'll review them here. And I just think that these were so interesting that these are the things that I wanted to bring to you tonight. In 2014, something really unfortunate happened up in Newfoundland, and that is that 9 blue whales died on the ice. They got stuck on the ice off the coast of Newfoundland and were killed. Now, most of the times when blue whales, which are the largest creatures that have ever lived on this planet, most of the times when they die, they sink. So very little, relatively speaking, is known about them. In the old days, when we used to throw harpoons at these things, and which was horrible, these were not considered to be the quote right whales, because if you threw a harpoon at them and killed them, they sank. So right whales were the whales that these were the whales that didn't sink when you harpooned them, so that you can haul them onto your ship. But in any event, my friends at the Royal Ontario Museum in Toronto, curators and scientific assistants, had been talking to the public and to media about whales for years and years, because they had a nice collection of whale bones there. So my friends would be asked questions like, what's the largest heart in the world? Blue whale heart. How big is it? It's the size of a sedan. But in reality, they did not know how large it was, because a blue whale heart had never been recovered before. So when these 9 whales died in 2014, fortunately, 3 of them did not sink. They got caught up on an ice floe, and they floated ashore. And 2 of them floated ashore in small fishing towns in Newfoundland. And my friends at the ROM got phone calls saying, these whales are up here and they look like they're in really good shape. Would you be interested in coming up and trying to salvage them? And so they said, of course. So they got a team together and they moved up into this region. We're talking about construction equipment here. We're talking about all sorts of different heavy equipment to be able to access the inside of these whales. Once they were able to sort of cut their way in, you had 4 people entering into the whale to push its heart out, which they severed between these ribs that they were spreading. So it was just a remarkable job that they did. Now, and here you can see the staff of the ROM doing their work on this. Now the amazing thing is that when the heart was freed up, and then when I saw pictures of the heart, there were some huge surprises that were immediately apparent. First of all, if you went into a butcher shop and you ordered a beef heart, it would sort of look like a human heart. It would sit there. It would have form, okay, shape. when I looked at pictures of the blue whale heart that they had pulled out of this whale, it reminded me of a 400-pound soup dumpling. It just collapsed into a blob, and it was a whole lot smaller than they imagined it would be. So it literally took them 5 years. It was a 5-year-long process. Here you can see them hauling the thing out. They had to they froze it for a year. Then they had to soak it to clean it. Then, you know, we've all been worried in biology class sometimes about splashing formaldehyde on ourselves and how dangerous that might be. Well, here they had to preserve this heart in thousands of gallons of formaldehyde. So the fear here is falling into a vat of it. And you can see that they're wearing a breathing system here so that they're not breathing in those toxic fumes. And they used to plug up these holes because you had to preserve the inside. You had to get the preservative inside the heart and outside. They used all of these different sized bottles to plug up the vessels so that the preservative could do its job. After this was done, they sent the heart overseas to something called a planetarium. And if any of you have ever seen the Live Bodies exhibit, it's basically, here's a guy who's dribbling a basketball, but he doesn't have skin and he's made of plastic. That maybe you've seen those things. Well, this was the largest specimen that the Grubner Planetarium in Germany had ever undertaken to preserve. And what they sent back 5 years after the heart was removed from the whale was absolutely spectacular. And here you can see that they have injected the blood vessels with different color latex so that you can see the difference between arteries and veins, and there are nerves in here. You can see the valves, which are the size of sort of a kid's tom-tom drum rather than a quarter. And the other thing here is that, look, it's bifurcated. It doesn't have that sort of pointy tip. And once again, it was a lot smaller than they thought it was, than they thought it would be. Now, the reason for this, they think, is that if this were a hummingbird that weighed 90 tons like that blue whale did, and they had it laid out next to the blue whale, and they looked at the heart of that hummingbird would be 8 times the size of the blue whale heart. And the reason for that is that a hummingbird beats its wings about 80 times per second. So in order to do that, it has to supply those wing muscles with a tremendous amount of nutrients and oxygen, produces a lot of carbon dioxide and a lot of waste material. So the heart is beating at 1,260 times per minute, which is a ridiculous number when you think about it. And we think that that might be about the physical limit, that if you think of the heart as a machine, that this thing can fill and empty and then fill and empty 1,260 times per minute. So now if you're going to get more blood to those to those wing muscles, and you're limited by now you're at the maximum rate that the heart can beat, the only other way to get more blood to those muscles is to have a larger heart. So in this case, relatively speaking, the hummingbird heart is 8 times the size of the blue whale heart. Blue whale heart beats maybe 15 times per minute, as opposed to 1,200 times per minute. And the other thing about it is that when the blue whale dives deeply, and we're talking about thousands of feet, its heart beats maybe 2 or 3 times a minute. And we think that what's taking place here is that remember the soup dumpling, that pressure, rather than sort of having a structure that is rigid and tries to, is under this stress of that tremendous pressure, it just collapses. And that's why the soup dumpling look. So there were all sorts of surprises that they ran into, and there are some blood vessels that they found that they still don't quite know what they are. Now, this is a mammalian heart, so you'd expect we know everything to know about mammal hearts. Just not the case. So a spectacular specimen, and it is now back on exhibit at the ROM until March 2022. If you get a chance to go up there, you will literally be blown away. But the size of this thing, originally they were going to have a big SUV sitting next to it, and now they've gotten something like the size of a golf cart. It is a lot smaller than they thought it was going to be, but it's still the largest heart in the world. Okay, the next story that I wanted to tell was about horseshoe crabs, and I know that you guys have got them on the Pacific coast, on in the Northeast where I live. Late spring, early summer, we see thousands of these things come into our beaches from the deeper water where they spend most of the year in order to mate, and they are literally, we toss this term around a lot, and that is living fossil, but horseshoe crabs have looked like horseshoe crabs for 500,000,000 years. To put that in perspective, that's 250 million years before the first dinosaur. Horseshoe crabs were creeping around the bottom of oceans. I wanted to look at them because if you remember back to that slide where I showed you the pacemaker, where the stimulus for the beat of the heart comes from within the heart itself. And I had this sort of snarky idea that I would show a very different type of heart, and sort of lead off with the fact that if you've ever seen movies where Aztec priests march, they march these prisoners up to the top of a pyramid, and then they sacrifice them and pull the heart out of this warrior, and let's show it to their followers, and it's beating. But this is why you would never see an Aztec priest holding the heart of a horseshoe crab, because as soon as you removed it from the horseshoe crab's body, it would stop beating. Now, yeah, so that's ridiculous, and that part didn't make it into the book, luckily. But I wanted to show a type of heart initially that was dependent on having the beat of the heart was dependent on having nerves that came in from the outside. There's no pacemaker. There are bundles of nerves called ganglia that attach to the heart, and they send the electrical signal that causes that heart to beat. So that's what I started out looking at. But what I quickly found out was that these creatures are in some serious, serious trouble. Now, beginning in the early part of the 20th century, and you can see walls that were basically built of these horseshoe crabs that were mostly recovered when they were coming in to reproduce. And these shots were taken in New Jersey, and they were used for fertilizer. And millions and millions and millions of these animals were killed. Now, luckily, people figured out that there was better fertilizer to use, and so that industry went crashed early 20th century. But more recently, fishermen who fish for eels and for whelk, kind of a snaily looking thing in a big shell, that they chop up horseshoe crabs and use them for bait in their traps. And this has put a dent on horseshoe crab populations. But the key problem that they've run into is that in the 1960s, a researcher figured out that horseshoe crabs, in their blue blood, which is I go into it, I explain a whole lot more in the book than I have time for here. But they isolated a substance in the blood of horseshoe crabs that, when it comes into contact with endotoxin, forms a visible clot. Now, let me back up and explain what endotoxin is. Endotoxin, I used to think, well, what is that? Is that produced by some bacterium and then released as a pathogen to hurt hurt people? No, an endotoxin is a substance that makes up the cell wall of bacteria called gram-negative bacteria, and there are a whole list of them. Now, the problem is that if there's a place where these bacteria exist, and let's say in a hospital room, and someone goes in and sterilizes that room, what happens is that during that sterilization process, the bacteria are blown apart by the sterilization. And those fragments of the cell membrane of the bacteria, if they get into the human circulatory system, they can be toxic. And those are endotoxins. So it's really a construction substance for bacteria, but when you kill them, it becomes deadly to humans. And this substance in horseshoe crab blood gives off a visible signal when it comes into contact with endotoxin. So once researchers figured this out, then an industry built up, and there are a number of different companies who during the mating season, when the horseshoe crabs come in to shallows, they collect these animals. And I live on the east end of Long Island. And I used to always wonder why I'd see pickup trucks just loaded. The back of the pickup truck is loaded with hundreds of horseshoe crabs. I had no idea what was going on. They're being transported to facilities that, as you see here, hang them upside down, stick a cannula, which you can see here, into their heart and drain their blood until it stops draining. Now these animals are transported to these facilities in unrefrigerated trucks, generally speaking, no water for up to 72 hours. They're bled up to 40% of their total blood. And then by law, they're released back in the wild. Well, many of them are dying, and we're in serious trouble because of that. And especially these populations. Now, that's not to say that thousands and thousands of lives have not been saved by this technique. So you know, I'm not trying to minimize the good that this substance has done, but we're driving these creatures to extinction. Now, there is good news, and this came about around 10 years ago. And through DNA technology, a test was developed to detect endotoxin without the use of any horseshoe crab blood. And a major drug company started using it. And the next step was going to be the mass production of this and opening up of a company to do this. The problem was that COVID hit. And when COVID hit, there was this need, you know, all of us are familiar with the sort of hospitalizations and everything that needs to be sterilized from medical equipment, respirators, everything you can think of that is going to come in contact with someone that needs to be sterilized. So they've fallen back on this old testing system and sort of pushed the new test to the back burner for a while. Hopefully, once we get COVID under control, if we do that, the horseshoe crab test will go the way of everyone's heard of the sort of the rabbit test that they used to use in drug companies. They thankfully don't use that anymore. And the other good news is that Cornell University came up with a synthetic substance that smells like horseshoe crabs, and they've been giving this out to eel fishermen and to whelk fishermen. And I did an interview last week, and the interviewer said they should call that stuff. They should call that stuff. I can't believe it's not horseshoe crab. I thought that was a brilliant name. But in any event, you could put this on toilet paper and stick that toilet paper in a roll of toilet paper into an eel trap, and the eels will presumably chase it down. So stay tuned. Hopefully, we'll be moving away from what you see here in this slide and using new technology to detect endotoxin. Okay, so one of the questions that I asked in this book was, where did this idea come from that the heart is the seat of the soul and intellect and emotion? And when I was writing a book about cannibalism, I was interested in why we had this knee-jerk reaction to the very word cannibalism. And I was going to title that chapter "Blame It on the Ancient Greeks." And by the same token, when I figured out where this idea of cardiocentrism came from, I was going to name that chapter "Blame It on the Ancient Egyptians." And the ancient Egyptians saw the heart. They saw that it was beating. That they saw that it was centrally located. That if you scared somebody, it beat faster. So it seemed to respond like a living thing did. So their idea was that this was an incredibly important organ that was involved in things like what we would consider the soul, intellect, memory, and emotions. Here you see the heart being kept separately, preserved separately in a little jar to be weighed against the feather of Mot in the afterlife. That's the goddess of truth and virtue, sort of trying to figure out, is there more good or bad that this person did? Or we're going to let a monster, which you see here, tear them up, or are they going to live in, are they going to have a happy afterlife? The thing is, though, that these ideas by the ancient Egyptians, and most of them with regard to the heart, were wrong. They believed, for example, that arteries carried air. And ancient Egyptian medical information was held in high esteem by the ancient Greeks. who picked up on this idea, not only of cardiocentrism, that the heart was the end all with regard to emotion, etc. The brain, they thought, was the Greeks thought that the brain was a radiator to cool the heart, and the ancient Egyptians didn't even care that much about the heart, about the brain. Well, when they mummified you, they would yank the brain out of your head with a hook through your nose. So they didn't think too much of it. But the Greeks, and after them the Romans, picked up on this idea of cardiocentrism, but they also picked up on the fact, on all of the things that the Egyptians got wrong. Now, and in the meantime, artists, poets, playwrights, painters are going, oh, the heart is the center of emotion, etc. And they are now responsible for the things that we still, artists are still doing, that sort of idea that the heart is the center of especially strong emotions like love and hate, and the soul, and all of these heartfelt and blue bloods, and all of these kind of connotations that we know about. Unfortunately, so there's nothing wrong with that. Especially when you think of some of the alternatives. If you were to put brain instead of heart, you can sit there and think of some really tacky sounding songs. But the main problem here is that the ancient Romans, especially an ancient Roman by the name of Galen, who was a surgeon, lived in the 2nd century CE, Common Era. He eventually wrote about 3 million words on anatomy and physiology and how the body works. The problem is that he did not have access to human bodies to do his dissections, and he fell back on what the Egyptians and Greeks had taught the Romans about the fact that arteries carry air, and also had this idea that, well, if you want to stay healthy, you have to balance these 4 humors: blood and black bile and yellow bile and phlegm. And if you do that, you can be completely healthy, whether it's mental health or physical health. And this is why for millennia, they would bleed people instead of giving you an aspirin and telling you come back in the morning. They would bleed you. Extremely problematic here, because after the fall of Rome, Galen's works, and I said 3 million words, were not initially translated into Latin, which was at the time the sort of language of scholarship, and sort of sat there. In the early Middle Ages, it was translated into Arabic by Syrian Christians, not into Latin yet. When it was eventually translated into Latin during the Middle Ages, it was the Arabic version with the sort of Christian slant that these Syrians had put on it. So the church came around and saw this translation, and they loved it because of the sort of Christian bent that the Syrian translators had given it. And so they declared that Galen's mistranslated work, most likely, and it was faulty to begin with, was divinely inspired. And for 1,500 years, more or less, in the West, the fields of medicine stagnated. Nobody could do any research. Everyone had to just fall back on Galen. And so this was this idea of the 4 humors. This was an idea that, for example, that venous blood on the right side would move to the left side. That nobody really knew at the time that on the right side, that blood went to the lungs and then back to the heart, to the left side. They didn't make that connection. They thought that somehow the venous blood would move through invisible pores and mix with this strange spirit-like substance in the air that was breathed in, called pneuma. So they got a lot wrong. And it stuck around that way for about 1,500 years. So you get things like, in 1799, George Washington is bled 40% of his blood after coming down with a throat infection, and he winds up dying the next day, going into hemorrhagic shock and dying. But this was not, these were not quacks who were doing this work. These were the top medical people of their day. who believed what they were doing was the right way to do it, and they were trying to save his life with the methods that were used at the time. And if you went 100 years into the future, from 1799 when Washington passed away to 1899, they would have been doing the same thing, only they would have been using leeches to bleed people instead of cutting them and bleeding them into a bowl. Okay, so when did Galen get the boot? About the 17th century in the West. Elsewhere, in places like China, India, and especially the Middle East, it was much earlier than that. But that got no airplay in the West. So in places like in Europe, and then in North America eventually. So William Harvey, who's really famous for being the one who made the connection between the right side of the heart and the left side of the heart. He's famous for that. And after that, you started to get all sorts of different innovations, and we were not falling back on this on a Roman who'd been dead for 1500 years, 1600 years. We started to get some real neat innovations that I talk about, and I don't have a tremendous amount of time to go into them, but the first stethoscope was invented in 1816 in Paris during a terrible outbreak of what they were calling at the time consumption, because it literally seemed to consume the body from inside. We now know that this is tuberculosis caused by a bacterium, and the problem is that in order to, one of the ways to characterize tuberculosis is that you could hear the liquid buildup in the chest. So physicians had to sort of put their ear to a patient's chest, and this is at a time when sanitation conditions were poor. Some people had parasites like lice. Other physicians didn't want to put their ear against the breast of a female patient. So this young physician was walking in the park one day and saw two kids playing with a stick, and one of them held the stick to his ear, and the other was sort of tapping on the end of the stick. And he got the idea to do this, a simple tube that was really the first stethoscope, an instrument that is still used today. The first cardiac catheter, which was invented in 1928, that is a story that, I write fiction as well. If you told me to write a bizarre fictional tale about how this came about, I couldn't come up with anything weirder than Werner Forssmann, who did this catheterization that you see here on himself after tricking a nun into letting him in late at night. She thought he was going to do the catheterization on him. He straps her down. He really just wanted to get into the cabinet because she had the key to the catheters, to the urinary catheters, and he used this on himself. He cut himself right above the elbow, and then pushed this catheter in. Took pictures of himself under a fluoroscope and saw that he had finally gotten this thing into his heart. He gets fired. He becomes a Nazi. He's in a concentration camp, comes out, becomes a lumberjack, and then 20 years down the road, he wins the Nobel Prize. So we're talking strange, seriously strange story. Okay, so I mentioned before that when I started to talk about when I started to get into the animals, that a lot of them tied into things that were related to medicine. And so you have, for example, the Antarctic icefish, which doesn't have hemoglobin. So it doesn't have the oxygen carrying compound that all other vertebrates have. What it has is, and how does this thing live in frozen conditions? So I go through a number of different very, very strange adaptations that it has. One of them is that it has antifreeze in its blood. And so I looked for an application that the antifreeze might be used for today. And what I came up with is that it's being used by ice cream makers to prevent, ever have ice cream and you eat half of it and then you put it in the freezer, and when it comes back out, it's all crunchy. That's because there's large ice crystals form. And this antifreeze prevents that because it latches onto the small crystals that normally form in ice cream and prevents them from becoming larger. So it improves mouthfeel. So I can tell you that that the other examples that I went over had a lot more applicability when it came to the field of medicine. One of them is, we finally convinced ourselves that the heart is not the center of things like the soul and emotion and the mind. And then in 1990, 30 women show up into hospitals with chest pains. When they do the workup on them, they can find no heart damage at all, no clogging of the arteries, nothing that has made them dizzy or given them these symptoms. What they found out was that all of them had, in the last month or so, had these horrible emotional events. They had people die, they lost their houses, they lost their jobs. And this is a story that I go into. The Japanese researchers called this Takotsubo syndrome, which means Takotsubo is octopus trap, and that's these pots that they use. The fishermen lower them down. The octopus thinks he's found a cool home, climbs in, and then they haul up the octopus, and then it's problematic for him. But in any event, in the West, we call it broken heart syndrome. And there's what seems to be happening is that when we go through stressful periods, our body pumps out stress hormones. Think of the fight or flight response. This causes our heart rate to go up, our blood pressure to go up, our breathing rate to go up as we're coping with some real or imagined threat. But that when the real or imagined threat goes away, that system turns back down. It stops producing these stress hormones. And what they've hypothesized is that these mostly postmenopausal women, 90% of them, that are exhibiting these the heart problems, heart pain, chest pain, dizziness, without having clogged arteries, for example, that their fight or flight response is not turning off. So this stress hormones are being released at a hyper rate for long periods of time. And that's causing coronary arteries that supply your heart. These are the things that usually clog, not to clog, but to spasm. And when they spasm, they're acting the same as if you had a clot. They're cutting off the blood flow downstream, which is a problem. So the tissue downstream in the heart, that whatever was supplied by that vessel can die. And in this case, it doesn't die because the spasm goes away. And then the good news for these folks is that it seems that within 3 to 6 months, this resolves itself. So, but the take-home message is that there is indeed a connection between the heart and the mind. It's just very different than the ancients believed. Two of the most fascinating stories were zebrafish, which are common aquarium fish. And I'll be brief here. I go into a lot more detail, and once again, these are neat figures by my artist friend Patricia Wynn. If you snip off 20% of a zebrafish's heart, the thing grows back perfectly functional. And this is something that's very different than what I just described. If you were to have a blockage of a coronary artery and the tissue downstream of that blockage dies, even if the flow resumes, you get rid of that clot, that tissue, when it heals, is not contractile anymore. It's not good contractile muscle tissue. It's scar tissue. So what is it about these zebrafish and their hearts and their blood that enables them to repair far more damage in their hearts than we are able to do? And there are researchers who are trying to figure that out and look at the compounds so that we may be able to take a major step in cardiac medicine. Burmese pythons are a real problem in places like the Everglades. Estimates that there are 1 million of these invasive species. Pet owners have released them over the years, and they grow to 20 feet in length. And so they're destroying all sorts of animals in the Everglades. Serious problem. So a researcher that I met was doing work on these animals and realized that when they had a large meal, that within a day or so, their heart grew 40% in size, 40%. Now, when we hear of enlarged heart, we think this is bad, but this is healthy heart growth. This is heart growth that you might see in an athlete, where the heart is getting bigger and stronger. This is not pathological. This is not bad. And so the idea here is that what is it about the python? What does it have in its blood that enables its heart to grow healthy or exhibit healthy growth without exercise? Idea here being this: if you've had a cardiac, some type of a cardiac problem, then they're probably going to put you on an exercise regimen to strengthen your heart. But there are some people who can't go on these exercise regimens. So because their heart is in bad shape. So might it be possible in the near future to use the compounds, and I discuss them in the book, that they've isolated from python blood and give them to these patients who are not able to do normal exercise in order for them to have heart growth and strengthen their heart after some type of a cardiac problem. This is now being worked on. Okay, this is the next to the last thing I'm going to talk about. This was absolutely amazing. So I went up to Harvard University and I talked to Dr. Harold Ott, and he's dealing with a problem that a lot of folks are dealing with, and that is that thousands of people die every year from waiting on lists to get a donor organ. And we're not just talking about heart, we're talking kidneys, liver. The problem is that you can't just take a heart and transplant it into someone's body, because unless the tissues match up, unless the blood type matches up, the heart, the recipient is going to reject, his immune system is going to attack that heart and reject it. So that's one of the key reasons why that we don't have enough hearts to go around for the most part. We don't have enough compatible, tissue compatible, blood type compatible other organs to go around. So what Dr. Ott wants to do, and what he's doing, is this. Let's say when you pass away, before you pass away, you are going to donate your heart, your body to science. Instead, he's going, no, donate your heart to this facility, and this is what we're going to do with it. So he's taking cadaver hearts and he's putting them through literally a detergent rinse that drips a detergent through the heart. And so he hangs these things up. And here you can see this micrograph shows what you see here in red is muscle tissue. And so the sequence goes like this. So, and here you can see that there's less muscle tissue as this process progresses, until at the end, all of the cells that your body would reject if you'd taken that heart and put it into someone, all of them have been dissolved away. And what's left is a connective tissue model. Most of this is collagen, and your body does not have this major rejection response to collagen. So what he wants to do then, and this technology exists, if you're the recipient, if you're going to be the recipient of a heart, right? He's going to take a sample of skin cells called fibroblasts and convert them into stem cells. We know that stem cells can be stimulated to become any type of cell, right? This can be done. This is certainly done in the body, but we can do this. Then he's going to take those stem cells and stimulate them to become cardiac muscle cells. Those cells now will match that recipient, the person who gave up the skin cells. He's going to culture those muscle cells and then seed them back onto this heart and literally grow a heart to order and then transplant it into that person. So I said to him, how long before this takes place? He said, within 10 years. So, I mean, that literally blew me away. So I talked more about that. And then the last thing I'll talk about this briefly here is that when blood vessels are transplanted, it's very difficult to transplant very small veins. Arteries, not so difficult because they're thicker. Their walls are made of muscle, but veins that are carrying blood back to the heart are very thin. So that makes them hard to knit together if you're moving one in, if you're transplanting one, let's say. So I went to see Dr. Glenn Gaudet at Worcester Polytech Institute in Massachusetts, and he came out and showed me he's trying to deal with this problem. And he came out with a salad bowl, and he said, "Bill, what do you see?" And I said, "Spinach." He goes, "Well, look closer." So I look closer. He goes, "What do you see?" I said, "Well, there are veins here." And he said, "Exactly." So he's taking spinach leaves, and you can see there's one inside this bottle here, but there's detergent up here, and he's doing the same thing. He's draining a detergent through these spinach leaves and dissolving away everything except what is literally the connective tissue of the plant world, so that what you see here, and here's one that is like half done. There's a vessel there, right? And that vessel, he's envisioning taking this thing and embedding human vascular cells, blood vessel cells onto this and growing these veins, and then being able to transplant them into humans. So these are the types of things that I found that connected modern technology and future technology with the animal kingdom and the plant world. And that's really what I wanted to do in this book. You know, this is not an encyclopedia of animal hearts, nor is it a textbook. I don't use jargon. I try to tell entertaining stories, use humor where where appropriate, and get some lessons in there about, for example, one that I've been interviewed a lot about that people are sort of surprised about is, if you're going to go out and shovel snow, you don't want to eat a big meal first, because to make a long story short, that blood is being diverted to your guts so that you're digesting your Cheerios, and it's not going to other places. One of the other places it's not going are your coronary arteries. So your heart is getting less blood because a lot of it is being shunted to your digestive tract. Now you run outside after a big meal and start exercising by shoveling massive amounts of snow, and you're putting your heart into a stressful situation, especially if you have if you've got heart problems to begin with. So those are the things that I'm interested in teaching my readers. You know, I stopped the book cold, and when I talk about carbon monoxide, and I go, look, if you don't have a carbon monoxide detector, just stop reading this book right now. And or if you know people who don't have one, go tell them to go buy one for them now, and then come back and pick this up. So conclusions: a variety of structures have evolved to supply oxygen and nutrients, and to get rid of carbon dioxide and waste products. And we need to not think about them as primitive or defective. Many types have evolved, especially in the invertebrates. They all work really well. The ancient Egyptians, if you want to see where the basis for this idea of cardiocentrism, that the heart is the place where emotions and the soul and the intellect and the mind, it was the ancient Egyptians, passed on to the Greeks, passed on to the Romans, moved into the Western civilizations. A real problem that I ran into, and it popped up in all three of my nonfiction books, is the sort of problem with lockstep adherence to the teachings of Galen, and the massive problems that caused in the field of medicine. And finally, medical advances are out there, and they're coming from every corner of the globe. There's a team in Israel who are computer printing hearts. So they're taking a different tack than Dr. Ott with his use of cadaver hearts. And so cardiac medicine is being transformed right now. So that's, to me, there's a lot of spectacular, cool stories to tell, and I try to tell some of them. So I want to thank the American Museum of Natural History and Long Island University Post for their support. I also want to thank the Seattle Town Hall and Third Place Books for having me here tonight, my editors and my publicist, and the great team at my wonderful publishing company, Algonquin. And finally, special thanks to Patricia Wynn for putting up with me for 30 years. And we're office mates at the Museum of Natural History, and as I said, she's worked on every single. She's worked on 250 different books. She's an award-winning illustrator, and I'm absolutely thrilled to be able to work with her. So, if you want to read more about me, Pump just came out 2 weeks ago, so it's available everywhere and in every format. And if you want to know more about me or contact me, you can find me at billschutt.com. Also, I'm on Twitter and Facebook. I've got 3 TED Ed videos. So if you want to start with one, the most recent one is about blood transfusions and how they began. These are my novels down here, sort of a World War II techno horror trilogy, cryptozoological books. And Cannibalism and Dark Banquet, like my other books, are pretty much still available wherever books are sold. So thank you very much, and I hope I left time for a couple of questions.
All right, Dr. Schutt, thank you so much. You covered a lot of ground there. That was very fascinating. you anticipated some of the questions, so I will leave those out. We are a little bit limited on time, so I've got 3 questions here that I think that that you maybe didn't quite cover, and I think are pretty interesting. So, the first one, I know that there have been human heart transplants. Have there been any other animal heart transplants that have been successfully performed that you know of?
Yeah, sort of the anti-Galen story that I get to tell in this book is about a baby Faye. And in 1984, Dr. Leonard Bailey transplanted a baboon heart into a human, and it had tragic results after 3 weeks. But because of, he did this not because. Well, let me back up. He started out by doing these xenotransplants. He would transplant the heart of a baboon into a sheep, and from one type of animal into another. And the main reason he wound up transplanting a baboon heart into baby Faye, who was desperately ill, was because there were no infant donor hearts at the time. And so I call this a sort of anti-Galen story, because tragedy was that baby Faye died, not because of a rejection of that heart, but for other reasons that I talk about. But the media covered it. There was such tremendous media coverage that a lot of people all over the world knew about this story. And after it was over, then donor hearts, infant donor hearts, started to come in. And Dr. Bailey did over 200 infant to infant transplants after that over the course of his career. They never had to do the baboon, as what are called xenotransplants from another animal into a human. But yeah, he successfully transplanted hearts between different animals, and that they went on and lived full lives. But yeah, and the other thing that came out that was good about this is that other researchers took this tact there, that they weren't. It wasn't necessary to. If baby Faye were alive today, they wouldn't do a heart transplant. They would do surgery on her heart. And so there are all sorts of methods now that have evolved, techniques that treat infant heart disorders that did not exist before baby Faye.
And that's probably just the less risky way to go about it, rather than completely doing a whole new heart.
Absolutely.
Yeah. Let's see. Is there anything that we don't know about the heart? What are we still researching, maybe about how it actually functions? Or
yeah, there's plenty that we don't know. And the key is to me, the key thing that is being looked at most closely is why we are not able to, after a clot has been broken up in a coronary artery, which there are 4 of them, and they supply the heart muscle with oxygen and with nutrients, and they're the ones that get blocked. And so the downstream of that block, that tissue dies. And the big unknown now is how to make that tissue, when it regenerates, turn into contractile tissue, good functioning muscle tissue, instead of scar tissue, which doesn't contract. So in a sense, people who have multiple heart attacks, it's like you've got this beautifully working pump, and different parts of it are becoming defective as heart attacks sort of accumulate. So that's a biggie. I mean, that's one that once they figure out how to do that, how to stimulate those cells to become stem cells, or to implant stem cells, and allow them to become muscle cells, there's a Nobel Prize written all over that, because that's huge.
Yeah, wow. This final one is maybe a little more personal. It must be very interesting to be working at a museum. I was asking my colleagues during your talk if they've been to that one, and it's an awesome museum. I love that one.
I'm
so
very,
this question,
very, very lucky.
yeah, it's such a great museum. So this question is, what does a day in the life of a scientist working at the American Natural History Museum look like?
Oh, well, so mostly right now, so I won't talk about myself. I'll talk about my friends that I go in and mostly interview now. So they spend a lot of time traveling. They travel around the world. They also, many of them teach. There is a graduate school in my department, at the Museum of Natural History. So students come in, they take students on. They're also involved in the collection. It is museums like the AMNH and the Smithsonian and the Royal Ontario Museum, a lot of people who are visitors just don't get to see the massive collections that are the science collections, the specimens that, for example, when I was studying vampire bats, I had access to specimens that were collected in the 1800s because they were preserved and curated and cataloged. So all that type of work, there's a massive amount of work that gets done behind the scenes. People see the exhibitions and they think that that's the primary job, but curators really don't do much of that. There are whole departments that do the exhibitions. But there's a lot of education of groups involved. There are, we give tours there, but we're also doing a lot of travel and collecting and observing and filming. And so it's spectacularly interesting, I think. And I've, as a research associate, I sort of have the best of all worlds because I don't have to go in and punch the clock. I go in when I want. I do the research that I want to do, and nowadays I'm mostly talking to experts about topics that I want to cover in my books. But yeah, it's a great profession if you can get into it. Starts out volunteer work always. Everybody that comes through the department is pretty much started as a volunteer someplace at some time in their life.
That sounds awesome. You're making probably a lot of us jealous who just go to work at a computer every day. But that's awesome. Well, thank you so much for this talk. This has been so fascinating. I want to thank the audience also for your questions. Thank you for watching. If you are interested in purchasing a copy of Pump, I encourage you to use the link in the chat. That's going to take you to our partner tonight, Third Place Books, so we can support a local store tonight. I hope that we can have you someday in real life again. But until then, I hope that you have a great night and best to you with the rest of your work.
Thank you very much. It was a pleasure to be here, and take care.
Thanks.